Now, Chapter 22 discusses the reproductive system. The first thing we have to discuss would be an introduction and what is the process of meiotic division? How does it happen? And this art was actually inspired by the meiotic division, and you can see more about the artist through this link. So what is the overall function of the reproductive system? Well, whether it's a male or a female, it is made out of the gonads, and the gonads are partly endocrine glands because they do secrete different hormones, and they also produce gametes, which are the sex cells. The overall function would be to develop and maintain the secondary sexual characteristics for the individual, as well as producing these gametes in order to produce offspring. Now, the meiotic division is the process of producing sex cells. Sex cells are also known as gametes, while the other cells in the body are known as somatic cells. The somatic cell has 46 chromosomes, and we would have to split those in half in order to form a gamete, so that at the end of the day, each gamete, whether it's a sperm or an egg, would have one set of 23 chromosomes, and we call that a haploid cell, so half of what the somatic cell has. So that when the sperm and egg unite at fertilization, we have restored the 46 chromosomes back, and that production of sex cells is known as meiotic division or meiosis. Meiosis happens in two successive divisions. There's the first meiotic division, followed by the second meiotic division. Before meiosis even starts, though, there is a process known as replication of the DNA, where, and I'm going to go to the picture to kind of explain that. So normally our chromosomes are made out of this one chromatid. Before meiosis starts, the DNA would have to replicate so that each chromosome now is made out of two chromatids that are united at the centromere. Now, the first meiotic division is basically going to, the cell will have all of the chromosomes lined up in the middle where we pair the identical chromosomes. So if this was chromosome number 5, though this would also be chromosome number 5. This is chromosome 6, so will this one be chromosome 6? Okay, the pairing of these identical chromosomes is known as pairing of homologous chromosomes. And then those homologous chromosomes are going to split apart so that we have two cells. Each cell will now have 23 chromosomes, but each chromosome has two chromatids. The second meiotic division would be splitting these two chromatids and separating them from each other. As you can see here in this image, where at the end of the second meiotic division, we would have 4 cells. Each cell has 23 chromosomes, and each chromosome is made out of one chromatid. So going back here, remember again, you have to start off the meiotic division with replication of the DNA, where now each chromosome is made out of 2 chromatids. Those 2 chromatids are known as sister chromatids, and they are both connected at the centromere. So at this point, our cells are diploid, where each of them has 2 sets of chromosomes in the form of 23 pairs. Now, the first meiotic division is going to separate those homologous chromosomes, which are the exact same ones, gene for gene, and result in haploid cells, where each cell has 1 set of chromosomes, but these chromosomes are replicated. They still have the 2 chromatids. And that is the role of the second meiotic division. We are going to separate these chromatids from each other. So we end up producing cells that are haploid, but now these chromosomes have only 1 chromatid. Now the steps of meiosis, the first meiotic division, it's made out of 4 phases. So are the second meiotic division, and they go in that exact same order. So there's prophase 1, metaphase 1, anaphase 1 and telophase 1. To remember them, they are past my anatomy test. So prophase 1 is very unique because it involves three different things. It involves synapses, which is the pairing of these homologous chromosomes. It also involves a process known as crossover. And I'm going to go here in order to understand the crossover and recombination. The whole idea behind synapses, crossover, and recombination is that to produce gametes that are not carbon copies of the individual, so that the offspring, although they might have characteristics of their parents, but we are not carbon copies of our parents, and we do not produce offspring that are exactly identical to us. So you'll see here the first step is synapses, where again the homologous chromosomes are paired. And then there's crossover, where now part of the chromosomes are actually crossing over each other. And then there's a process of recombination. And in recombination, you will see that part of the genetic material is exchanged between the chromatids. So you see here this one chromosome that used to be all light blue and this one that is all dark blue. You see that part of the genetic material was exchanged. And that again would produce gametes that are unique and are not carbon copies of the parents. So that's what happened in prophase 1. Again, very unique to prophase 1. It does not happen in prophase 2. So after that, the cell will move on to metaphase 1, which is, metaphase means that all of the chromosomes are going to be in the middle of the cell. So M with M. The chromosomes line up in the middle, and they are going to be connected with these separated apart by these spider webs. These spider webs are known as the mitotic spindles, and the mitotic spindles are connected to these centrioles. Now the mitotic spindles are going to pull these chromosomes apart in order to make these two cells. So metaphase would be the chromosomes lining up in the middle. Anaphase would be pulling the chromosomes apart, again, which is the function of these mitotic spindles. So anaphase, apart, A and A. And then telophase, which is the last part of meiosis 1, you'll have that each cell, that cell now has to totally separate from each other. So there is a process of cytokinesis, which is separation of the cytoplasm and producing these two identical cells. We talked about the first meiotic division. Now, second meiotic division begins right after telophase 1, and you have the exact same 4 phases in the exact same order, and they are just we just call them prophase 2, metaphase 2, anaphase, and telophase 2. Now at the end of telophase 2, we would have 4 gametes, or we'd have 4 cells, and each cell has 23 chromosomes, 1 chromatid each. In the process in males, we produce at the end 4 sperms, but in females, we produce only 1 egg. The remaining 3 cells degenerate. Okay, so at the end of spermatogenesis, which is the process of producing sperms, this ends up making 4 sperms, while in the process of oogenesis, which is the production of eggs, there is only 1 mature egg that is formed, while the other 3 cells, which are the end products of the meiotic division, those are going to degenerate. And we'll talk a little bit more about that when we get to the male and the female reproductive systems. So the division ends with one set of each cell has just one set of genetic instructions, or just 23 chromosomes. Again, when it unites with the other gamete, we will go back to what our somatic cells have, which are the 2 sets or 46 chromosomes. And you can see here the process of crossing over that happens during meiosis 1, where we are making 4 unique gametes at the end. So the crossing over provides for a unique combination of traits in our offspring. In the next part, we are going to start talking about the male reproductive system, and we'll talk about the gonads and the spermatogenesis process.